So we're going to look at pretty much the history again of how they figured out how DNA replicates. So after Watson and Crick figured out the double helix structure, there were 3 ideas of how DNA replicated: the conservative method, the semi-conservative method, and the dispersive method. The conservative method said that parental DNA remains together, and then newly formed daughter strands are then together. So that would be this first section over here. So your gray is indicating parental DNA. So you had the parental DNA staying together, and the blue indicating the newly synthesized DNA. The second is in the middle over here, and this said that each of the 2 parental DNA strands act as a template strand, and so your new molecules of DNA will have 1 parental strand with 1 new strand. So it's semi-conservative since it's half and half, split half and half. And then the dispersive method suggested that both copies of DNA have double-stranded segments of parental DNA, and that newly synthesized DNA is interspersed. So old, new, and so it's kind of chunked and pieced together.
So the gray is the old?
Yep, the gray is the old. Think of it like old parents, gray hair kind of thing. So Messelson and Stahl investigated DNA replication with E. coli. So remember, E. coli is a bacteria. And so again, there's this lengthy process of what they did, and there'll be a video at the end to kind of help illustrate all of these words. So they used a medium containing a heavy isotope, specifically nitrogen 15. So a medium is something to grow bacteria on. And so this particular medium had the isotope nitrogen 15 in it. That nitrogen 15 then gets incorporated into the nitrogen bases of DNA, and as DNA gets synthesized, that nitrogen 15 will eventually show up in the DNA of the E. coli. So once he placed the E. coli in the medium after he did the N15, he let it grow for a few generations. Then he took E. coli cells from that nitrogen 15 medium and placed it into a medium containing nitrogen 14. Nitrogen 14 is what we call the normal form of nitrogen. And then again, he allowed that E. coli to grow for many generations. Once he stopped letting it grow, the next step was to harvest cells and isolate the DNA. Then he centrifuged it at high speed. So a centrifuge is a laboratory instrument device, and you take a DNA sample, you put it in a little tube, and then you stick this tube into this centrifuge machine. And if you remember what a rotary phone looks like, way back in the day where you had to put your finger in it and turn the dial, imagine where you'd put your finger.
It spins it.
Yeah. So a centrifuge then spins this DNA. So the DNA was loaded into a gradient with either cesium chloride or sucrose. He spun it at high speeds, around 50,000 or 60,000 RPM. And what he found was that, or what they found, sorry, it was a couple of them, that DNA formed bands according to its density. Since N15 has one more neutron to it, it's going to be heavier or more dense than DNA that has N14 in it. And so it would be lower in the medium than the N14, right? N15 would be below N15. N15 will be below N14. So what he found was that DNA grown in N15 forms a band at a higher density position. Remember, higher density literally means lower in the tube, okay, because high density you sink. Then further down the centrifuge tube, then the DNA grown in N14. After one generation of growth in N14, after they had been shifted, it starts to have a band observed as an intermediate position. Again, the video will highlight this much better than these words. So basically, with purely N15 grown bacteria, your band is down here, okay, in the red. After it was incorporated with the N14, it would be split 50-50. 50% of the DNA had N14 in it and 50% had N15. After it was allowed to produce another generation, there was 75% of the DNA had N14 in it and only 25% had the N15 in it. So this suggested that either semi-conservative or dispersive mode of replication was occurring. So it ruled out that fully conservative method in the beginning. So DNA harvested from cells grown for 2 generations in N14 formed 2 different bands, one at the intermediate position and one that corresponded to the band of N14. So that led them to believe that one strand of the DNA was completely composed of N14 DNA, and that the one at that intermediate position had a combination of both N14 components and N15 components to it. This confirmed the semi-conservative method of DNA replication. And then during DNA replication, each of the 2 strands make up a side of the double helix. They serve as a template for which new strands are copied. The new strands will be complementary to the parental strand. So if the parental strand had an A in position 1, the new strand would have a T in position 1. So that's what it means by complementary. It wouldn't be identical because the A can't pair with another A. It has to pair with a T. Okay?